And now we will hear from Dr. Thomas Finn. Dr. Finn is a CMC reviewer in the Division of Cell and Gene Therapy within the Office of Tissues and Advanced Therapies at FDA CBER. He joined the FDA in 2006, where he reviews regulatory submissions at all stages of product development in the cellular therapy field, from pre-IND advice to post-licensure supplements, and he performs pre-licensure and biannual facility inspections. He has regulatory experience in bioassays, product comparability, product stability, and process validation. He is also involved in the development of several guidance documents and numerous CBER internal working groups, such as CBER's CMC Coordinating Committee. His presentation today is about regulatory considerations for FBS, human serum, and platelet lysate used as reagents in product manufacturing. And we are delighted to welcome Dr. Thomas Finn.
Hello, my name is Tom Finn. I've been a CMC reviewer at FDA in the Division of Cellular and Gene Therapies for the past 15 years. Our division regulates a wide range of regenerative medicine products, many of which use serum reagents at some stage in manufacturing, either directly in the manufacturing process or as part of QC testing or product development. Because serum reagents can be considered critical reagents, they should be appropriately qualified for safety and performance. Serum reagents, like many reagents, may be difficult to choose and qualify. Cell and gene therapy manufacturers often struggle with trying to identify the right material to use and figure out ways of appropriately qualifying it so that they can establish that it's suitable for the intended purpose. Likewise, suppliers do not always understand how serum used as a reagent. What are the expectations for qualifying that for use as a material in production of a clinical product and for licensure, as well as all the different ways that serum can be used in the manufacturing process. And with that, I will begin my presentation. I'd like to start out by thanking the organizers for including FDA in this meeting. I hope to provide some FDA perspective on some of the considerations we have for serum reagents. And those include fetal bovine serum, human platelet lysate, and human AB serum. As an overview, I will go through some recent trends that we've seen, ways that you can communicate FDA information to FDA, our regulatory framework and our general regulatory concerns about these types of materials, our current regulatory approach, and also we'll discuss some thoughts on GMP grade reagents. Some recent trends in cell and gene therapy manufacturing are that, first off, we see that more developers are switching from FBS to human AB serum or platelet lysate, but FBS is still very commonly used. There's more use of serum-free medium, which typically contains human or animal proteins such as insulin or transferrin, albumin, et cetera. There is less use of human and animal feeder layers to grow cells. They're relying more on cell substrates and culture medias. There's an effort to reduce cost of goods, and that affects decisions on choices of materials, and a change in a critical reagent needs to be well managed as cells are sensitive to growth conditions. Some more trends. Late stage product development often leads to a need to increase product yield. So there's a greater emphasis on the ability and the performance of serum to meet that demand. Historically, most serum and serum reagents that were available were simply marketed as for research use only, although now we're starting to see some materials being labeled as suitable for cell therapy manufacturing. We also note that there are GMP grade versions available by some suppliers. But surprisingly, we don't see a lot of performance qualification by cell and gene therapy manufacturers themselves. They seem to be relying on whatever qualification the supplier is doing in order to, provide a consistent product. Ways FBS can be used could include, as a reagent in product testing, as a reagent for producing the products, for preclinical testing in animals, or for the actual manufacturing of the clinical product. And that can include, just being used as a way to increase the stability of the source material or an intermediate. It can also be used as an excipient for final product formulation for administration to the patient. So along with the drug that's being administered, the serum reagent may also be administered. Who's responsible for the safety and quality of reagents? Well, ultimately it is up to the IND holder and the BLA applicant, that is the manufacturer of the cell and gene therapy, that are responsible for all aspects of manufacturing. That would include reagents. Safety and performance testing can be conducted by either the supplier, the manufacturer, or both. When the supplier doesn't perform the testing, then the obligation is usually on the end user. And often we find that information that is provided by suppliers to cell therapy manufacturers and then communicated to the FDA is often insufficient or vague. And I'll give some examples at the end of the talk about what I mean by this. So ways to communicate with regulators, FDA, is to directly provide information in a regulatory submission. So that could be an IND or BLA or some other type of file. The description of something could be, something produced in-house or provided by a third party. Often that is, comes in the form of a certificate of analysis, but that information is directly embedded in the IND. Or we can get information through a master file, which is submitted by the supplier instead. So we'll talk a little bit more about how that works. Certificates of analysis are the most common means of communicating information about the serum reagents. But there can be vast differences in the level of information that is provided. It can be just a paragraph or many pages. Sometimes these certificates of analysis are rather vague, or they provide incomplete information. And there's a lot of variation in the level of testing that's performed. And then sometimes they're marketed as GMP grade. But that can help. That can provide some level of confidence. But without information about what that actually means for that particular material, it's not always terribly helpful. So let's go over those communication pathways in a little more detail. So if you are a cell and gene therapy manufacturer, you can get information from the reagent supplier. It could be a COA or other types of information. That information is then embedded in the IND submission, which is passed on to the FDA. In that submission, we will review it. If we see something that is of concern or there's missing information, we would reach out not to the reagent supplier. We would reach out to the IND holder and say, hey, there's information that we need. You should reach out to the reagent supplier. And then they would provide that information and it would come back in as a communication from the IND. We cannot share confidential information with reagent suppliers. In fact, we can't even acknowledge that an IND exists that might be using it. Likewise, information that the reagent supplier might be willing to provide to the FDA, should be communicated directly through the cell and gene therapy manufacturer, through the IND, not emailed to us. We don't really have a good mechanism for that to work. And it's really the responsibility of the cell and gene therapy manufacturer to be managing that information and to be doing risk assessment and deciding what information they need and how they're going to qualify a material. The other pathway then is this master file. Master files have the advantage that there might be information that a supplier doesn't want to share with an end user. Either it's too much information or it may be trade secrets. So one way of handling that is to have a master file that the supplier is the master file holder of, and they will, include detailed information, provide that, as a cross-reference, and we can then use that to support the IND. And so the cell and gene therapy manufacturer, that is the IND holder, would cross-reference your file, and then we would then use that combined volume of information to make a decision about that product. We cannot share information between the two parties. Sometimes that leads to some rather awkward discussions because we might identify something that's missing, it's not in the master file, or there's a problem with it, but we can't really tell, at least in any detail, the IND holder what the problem is. We have to discuss that directly with the master file holder. Likewise, we can't give detailed information about what the cell and gene therapy manufacturing is actually doing with that reagent. Master files do have uses, but the content is really up to the master file holder, and there's no obligation to have a master file. That's completely up to the decision of the supplier. The existence of a master file is no guarantee of quality of the reagent or compliance with FDA regulations. And that's because, for one thing, we don't approve or disapprove master files like we would INDs or BLAs. So whatever material, information, a master file holder wants to include in that master file, they can put in there. If there's problems, we will try to, work with the master file holder to help resolve some of those issues. But either way, it doesn't go on hold. It would still remain active and could be cross-referenced, but it wouldn't be supportive. It's not always clear to regulators exactly what is being referenced in a master file, because again, they can contain large volumes of information, and it's not always clear why they're being cross-referenced. Master files are not typically reviewed until cross-reference. And that's kind of an important point, because if you submit a master file to the FDA, but no one actually uses your master file, they don't cross-reference it, say, for 2 years, that means for those 2 years, you would have, that file would not be reviewed. So if you haven't heard anything back from FDA for 2 years and think everything's fine, it may not actually be that the case. So due to resource limitations, we typically don't review master files until they actually come in. So what are some expectations for reagents used in cell and gene therapy manufacturing? Ours is a risk-based approach. So basically we consider how something is being used, and then that kind of dictates the level of review that we do and the regulations that apply. So the highest risk situation would be in a case where you have an actual active ingredient that's meant to provide some medical benefit. And then of course you would need a clinical trial, performed under IND, and ultimately you would submit a BLA for licensure. So from a manufacturing standpoint, we would be paying a lot of attention to materials and facilities and processes. And ultimately, our concerns would be about the safety and efficacy of the product, and product quality and reproducibility of the manufacturing. Now, if a material is used not as an active ingredient, it's used as a reagent, and it has direct product contact, that would have a high level of risk, but it would be lower than for an actual active ingredient, and there would be different regulations that would apply. It could also have direct patient contact if it's an excipient, or if there's residuals left over from the manufacturing process. And so we would consider that. Some materials would have no direct product contact. For example, they might be used in an assay. That's definitely a lower risk, but still not risk-free, because the assays might be, really quite important, and the particular, serum reagent might play a very important role in that in terms of the actual readout that you get. So we would pay attention to the suitability and the performance of that reagent. Now, you might think that preclinical product manufacturing wouldn't be an issue, but there's still some risk, in the sense that we still pay attention to what materials are being used, and ultimately how representative that preclinical data is of the actual clinical product. So if somebody's using a lower grade reagent or a different serum reagent, we would have to consider how useful that preclinical data is. In considering, say the difference between the regulatory approach for, things like animal-derived reagents versus human-derived reagents, there's actually a lot of commonality. I mean, obviously there will be some differences, but in all cases, we're interested in the source of the animals as well as the source of the human donors and the country of origin in each case. We're very interested in blood donor screening and health medical questionnaires and testing. We're as well as we are interested in the health of the animals that are used to provide fetal bovine serum. In both cases, we're interested in the facilities that are used for collection and processing. We're interested in details about pooling or retests on certain pools, on whether something is filtered or not, whether there's mycoplasma, endotoxin, or sterility testing. And so lots of commonalities. There are some specific considerations for certain products, and we'll get into some of those. So at a kind of a high level, some of the things we think about when we think about ancillary materials in general is, is this a research grade reagent? Now, often the packaging or the COA says, not for clinical use, for research purposes only. So it can't be used, right? Well, of course, it can be if it's properly qualified. That's not always achievable for some reagents because they might be really only suitable for, experimental research and not actually for use in any clinical product production. But many research grade reagents are high quality and can be used. Sometimes additional testing is required. Human-derived materials, generally we're advising if there is a licensed version available, we recommend using that because in general, we recommend using the highest quality version of a material that's available. But if it's not licensed and they're generating it themselves, then we definitely need information on donor eligibility because we are interested about infectious agents, as well as other issues that we'll go over. And of course, for animal-derived materials, adventitious agents, so bovine viruses as well as prions. So again, the country of origin is very important. Serum-free medium does not always address all the issues. Some might think, some cell and gene therapy manufacturers might think that if I use serum-free, I'm good. Not always. For one thing, a lot of serum-free media supplements or serum-free mediums actually contain serum, animal or serum, or human serum components, such as insulin, albumin, transferrin, et cetera. Human proteins can either be recombinant or purified from source plasma, but each has its own safety concerns. And there's not always licensed versions of every material available. In terms of suitability materials, the most, I'd say, applicable regulation for reagents in general are GMP regulations, which are meant to apply for appropriate qualification of a reagent to mitigate risk and ensure an adequate level of quality. We're especially concerned about materials that have direct product contact, glass, tubing, cells, and so forth. In the case of serum, platelet lysates, and so forth, one of the main concerns, again, is infectious agents. But we're also concerned about lot-to-lot variation and material performance. In terms of research-grade reagents, again, that's a very broad category. And so we don't really think so much in terms of, well, what's research grade, what's GMP grade, what's a premium grade. We look at the details, what is the level of characterization, what actual testing is done, what level of documentation, what kind of, facilities are used and traceability and so forth. So it's not really about the actual designation of the material. But of course, some are now providing GMP versions. And of course, we for all reagents, encourage the use of the highest grade available. So is GMP always the highest grade? So first, let's consider, what does GMP really provide for you? It's intended to provide an appropriate level of oversight, intended to control reagent quality and safety, minimize contamination, cross-contamination, and manufacturing consistency. There are certain elements of GMPs that apply for any type of thing that you're making, whether it's a reagent or a product. That would include well-defined written procedures and adequately controlled, equipment and manufacturing environment, the building and the air handling, and accurately and consistently recording data, and that would include product testing. Now you'll notice in those three elements, facility, the actual physical building is only one of them, and well-defined written procedures is at least as important. So, and that's all part of a quality system. So in addition to knowing about equipment and buildings, we need to know about the actual quality system. And this information isn't always clear, especially if something's just labeled as GMP grade, without providing details about what that actually is. So we often see situations where, manufacturers want to, consider using a GMP grade reagent, but they wonder, is that really a requirement? They have to use it. And one of the things we ask is, well, what is the difference? What is the supplier telling you about, their different grades? And often that information is not clear. GMP is not a grade because it's not like applying to some or adhering to certain standards. It's a set of practices, and those practices will vary, as well as the building designs and how they're operated will vary depending upon what's being made. So just calling something GMP, it's not clear necessarily what that means. The fact that a reagent was made in a GMP facility does not necessarily guarantee that adequate safety and quality are in place, or that the material produced is appropriate for manufacturing a cell and gene therapy product. Again, the devil's in the details. FDA does not verify the grade of reagent, whether it's be research grade or whether it's GMP grade. We don't inspect reagent facilities. We do inspect manufacturing facilities and QC labs that are directly associated with the release of that product, but not reagents. So we rely upon the information that's in the IND about how that's produced and the level of safety associated with it. For bovine-derived materials, of course, in terms of mitigation of risk, as I've already said, traceability is important and verification of that. Of course, adhering to testing requirements for bovine viruses. If there's information provided about gamma radiation or heat inactivation, if used, that can be helpful. And some suppliers perform additional testing beyond what's actually required. Human-derived materials, again, a lot of emphasis on, on information being provided in the certificate analysis on donor testing and country of origin and any characterization that is performed. And we do like to see that in the case of human AB serum, that information on where the blood or plasma is collected and processed be provided, and the processing should be from, for blood and plasma collected at FDA licensed facilities. Although raw materials are derived from human origin, are not considered, I'm talking about serum, are not considered human cells or tissue products or blood components, according to the regulations. We recommend that people provide, and adhere to kind of the same level of donor eligibility expectations as you would if you were actually manufacturing a blood product or a cellular therapy, because the risks are essentially the same. Important consideration. So one note about source plasma. Sometimes we will see, in terms of describing the source, where the plasma came from, it'll say, oh, it's source plasma. Well, source plasma means something very special to FDA, because source plasma is a licensed product. And source plasma has its own specific set of regulations. So we know exactly, what source plasma is and how it's supposed to be tested. It's not always clear that, a supplier is really using true source plasma versus just plasma derived from, a serum that is derived from a plasma source. Also, there can be confusion about what heat inactivation means. So for a licensed blood product like albumin, the final product undergoes 10 hours of heat treatment at 59 to 60 degrees Celsius. whereas typically heat inactivation of serum for culture might only be 30 minutes. Now, each provides some level of viral reduction, but not anywhere near the level that is used for actual source plasma. Platelet lysate, our concerns about that are, well, the same as they would be for blood products or serum. But I should note that there's a lot more variability into what the source material could be. That could be whole blood, it could be platelets, it could be platelet-rich plasma, it could be fresh or expired platelets. Lots of variation in the actual method of processing it and preparing it and the equipment that's used. Differences in, any viral or pathogen reduction, pool size, just a lot of information about platelet lysate that's a little more variable than I think some of the other products are. I'll kind of end on the discussion of pooling. So pooling has both, positive and negative aspects. So on the positive side, pooling is a great way to help minimize lot-to-lot variation between different donors or different animals. And the larger the pool, the more likely you're going to have similar properties as the last batch of serum. The one disadvantage of that is that the information about the batch size or batch variability often isn't communicated to us, and that could very much depend on the particular supplier. So that would be useful information to know. On the negative side, the larger the pool, the greater the risk of an infectious agent that was not detected in an individual human donor that might be present in the pool. The larger the pool, the more likely you are to exceed the sensitivity of the assay. So one always has to consider the sensitivity of the assay if you're testing pools. So let's go through some examples and finish with that. We'll see situations where documentation will say, oh, this serum product complies with FDA regulations. Okay, well, that's great, but which regulations are you referring to? Are you referring to GMPs? Are you referring to biologics? Are you referring to blood regulations? So it's unclear. They might say, well, no, it's we are testing according to FDA regulations. Okay, that's great, but that's donor testing. What about donor screening, which is also important? Or they may say, hey, we're complying with donor screening and testing, but they don't give any information about, the actual test kits that are used. Are they using, cleared FDA licensed antibodies? What about antibody versus nucleic acid testing? In some cases, both are required. What testing facilities were used? What assays, were these validated? Were they developed in-house? We've already talked a bit about GMP grade and what that might mean, or lack of information about that. We've seen situations where COA will say on a viral test for informational purposes only. Okay, well, what happens if that comes back positive? And somebody might say, oh, well, if it's positive, we won't use it. Well, in that case, it's not really for informational purposes only. That's a release test. Virus testing, tested, might infer that all the testing is done, but that's not necessarily the case. Often when we look to see, well, is everything that's being tested for really being performed? And in some cases, the answer is no. There's issues about defibrillation steps because sometimes that can include bovine thrombin. So now you have the whole issue about bovine reagents as well, even though this is for a reagent. It'll say performance tested, but not really give much details about how that was tested. Is it just a cutoff? Is it a yes, no? Do you try to keep it in a particular range? Again, we've already talked about pooled. It would be useful to know what that actually means. And even for purified growth factors, you might think, well, there wouldn't be much level of risk associated with that. But sometimes, somebody will go to a great trouble of purifying a growth factor or a serum protein, and then they add bovine serum albumin as a carrier. Well, then there's no information about the BSA. And then a very common one is, well, this FBA is from supplier X, but supplier X has lots of different versions and grades of FBS. So which one do they use, which lot number and stuff. And so those are the kind of information we need. So let me just summarize by saying that FDA's review process is based on an assessment of risk. How a material is used may impact which regulations apply and to what degree. And for human AB serum and platelet lysate used as reagents, we recommend following the same donor screening and testing regulations that apply for actual licensed, blood products. FBS is widely used, but the level of information, as with platelet lysate and other materials, is often, highly variable in terms of the kind of information that's communicated. And sometimes that's insufficient. We are interested not just in the safety testing, but also in performance testing. That's not always performed by suppliers, but do realize that end users, that is the manufacturers, may not be doing any testing, themselves to ensure that it's giving a consistent result. GMP versions of materials can provide a higher level of quality, but we kind of need to know what that designation means, in their mind. And we recommend that product developers work closely with reagent suppliers to make sure that regulatory concerns are addressed. And with that, I will end and be ready to take some questions. Thank you very much.